Hello and welcome back to Cells and Movements Across the Plasma Membrane. This is a large chunk of lecture material that's been separated into 4 different segments, and this is segment 3 of 4 segments. In segment 3 of 4, we will finish transport mechanisms, and remember that means all the ways in which something can cross the plasma membrane. And then we'll talk about a new concept, but a very important concept, especially for the next chapter, called membrane potential. So you've seen this information before. I hope you remember that all transport mechanisms are divided into these 2 categories, passive transport and active transport. And passive transport, which we have recently covered, includes methods of crossing the plasma membrane that do not require the cell to spend ATP, and they rely on an existing concentration gradient. So for both versions of diffusion, simple diffusion and facilitated diffusion, as well as osmosis, those substances move because a concentration gradient exists, and because one way or another, the plasma membrane allows that substance to come through. Today, we'll wrap up transport mechanisms by talking about mechanisms of active transport. And active transport differs from passive transport in one principal way. That is, during active transport, the cell needs to spend ATP. The cell needs to spend its own energy in order to move something across that plasma membrane. And we'll talk about 3 types of active transport: primary active transport, secondary active transport, and vesicular transport, or transport in vesicles. During primary active transport, a transmembrane protein, often referred to as a membrane pump, uses ATP to move something across the plasma membrane against its concentration gradient. And when we say against its concentration gradient, we mean that the substance is moving from an area of low concentration to an area of high concentration. So in the image shown on this slide, where that vertical black line represents the plasma membrane, the red arrow represents the transmembrane protein that we call a pump. That pump will take the substance, represented by plus signs, and the pump will take that substance and move it from an area of low concentration to an area of high concentration. And as we'll see in just a moment, that red arrow, or that transmembrane pump, will cleave a molecule of ATP and use the energy that's released in order to drive the movement of that, in this image, that red plus sign, from the left side to the right side, from low to high. Here's an example of a membrane pump. In this case, it's a hydrogen ion pump, also known as a proton pump. I hope you remember from chemistry that a hydrogen ion consists of just a single proton in the nucleus. There are no electrons. So we can call this either a hydrogen ion pump or a proton pump. Notice that this pump is a transmembrane protein. It spans the entire width of the plasma membrane to contact both the intracellular fluid and the extracellular fluid. And this pump is going to move hydrogen ions from an area of low concentration to an area of high concentration. And in this scenario, that hydrogen ion is going to move from the cytoplasm to the extracellular fluid. So this pump is responsible for expelling hydrogen ions from the cell. Notice also in this image, there is a molecule of ATP. So pumps are also ATP aces. And I hope you remember that ACE often refers to the fact that a protein is an enzyme. So an ATP ace is an enzyme that can cleave ATP. So built into this proton pump is an ATPase. Part of this protein molecule acts as an enzyme that cleaves ATP into ADP. And the energy that is released drives a conformational change in the proton pump. In this protein, it changes shape as it picks up a hydrogen ion on one side of the plasma membrane and deposits it on the other side of the plasma membrane in an uphill fashion, meaning from an area of low concentration to an area of high concentration. We can use this image to compare and contrast two mechanisms of passive transport with the mechanism that we're currently talking about, which is primary active transport. So moving from top to bottom, the top two mechanisms are passive because the cell does not need to spend any ATP. And notice that the substances move downhill or along their concentration gradients from an area of high concentration to an area of low concentration. So up top we have those brown circles, those brown dots, moving via simple diffusion. So they move directly through the phospholipid bilayer. So they are necessarily small and uncharged and lipid soluble. And next to that, for facilitated diffusion, we have those yellowish triangles. Facilitated diffusion also moves substances downhill or down its concentration gradient from an area of high concentration to an area of low concentration, only now a membrane protein is required, a transmembrane protein. And looking below at that oval purple transmembrane protein, that's a pump, and it's responsible for active transport. Notice two things. Number one, it's moving a substance uphill or against its concentration gradient. It's moving a substance from an area of low concentration to an area of high concentration. And here, obviously, we're talking about the green squares. So pumps move things uphill or against their concentration gradient. And also notice that a molecule of ATP is required in order for this pump to work. This pump is a protein. It will change shape when the enzyme part of this protein cuts apart a molecule of ATP. That ATP becomes ADP, and energy is released in the process. That energy that's released causes a conformational change, causes a shape change in the protein, and that's what allows this pump to move the green squares from one side of the membrane to the other. We'll walk through this detailed example of the most abundant pump in your body, the sodium-potassium pump. The sodium-potassium pump is present in every cell in your body, and it plays a super important role. Lots of your ATP every day, a huge proportion of your ATP every day goes toward fueling these pumps. These pumps are responsible for maintaining concentration gradients across the plasma membrane such that if we look at any cell in your body, there is more potassium inside the cell and less potassium outside the cell. And if we look at that same cell, there's very little sodium inside the cell and there's much more sodium outside the cell. So pumps are also referred to as ATPases, meaning they're enzymes that have the ability to cut or cleave ATP into ADP, releasing energy in the process. And the energy that is released drives a shape change in the pump, a conformational change in the protein. So let's work through this image from the left side to the right side. And starting all the way on the left, I would like you to notice that the artist has drawn two arrows here to represent concentration gradients for two different solutes, sodium ions and potassium ions. And the artist has used the saturation of color to help convey or help inform you about the concentration of each of these ions on either side of the plasma membrane. So let's start with sodium. So notice here that the dark green color shows us that there are more sodium ions outside the cell compared to inside the cell. So we go from dark green to light green. The opposite is true for potassium ions. There are more potassium ions inside the cell, shown here in the dark, more saturated orange color, more potassium inside the cell when compared to outside the cell. So starting all the way on the left side of this image, we see that there are two different concentration gradients for two different ions. There's more sodium outside the cell and more potassium inside the cell. And the sodium-potassium pump, its primary responsibility is for creating this concentration gradient, these concentration gradients, and for maintaining them. And these pumps work 24 hours, 7 days a week. Okay, so let's move over to number 1, so we're moving over from left to right, where we see our transmembrane protein. It's called a sodium-potassium pump. It's purple. It's also known as a sodium-potassium ATPase. It changes shape, and the artist has taken care to represent that very carefully. Although this is an artistic representation, this is not actually what the protein looks like. I think it's a very fair and useful representation. So let's start with number 1. Where you see the number 1, the protein, the pump, has a particular conformation, has a particular shape that allows 3 sodium ions to enter this particular site, this nook or this cubby. And when those 3 sodium ions fit into their spaces within this purple protein, that allows the protein to then become an enzyme, a functional enzyme. So the protein changes shape ever so slightly after those sodium ions enter, and that activates the enzyme portion of this protein. And so now we're moving from 1 to 2. And in panel number 2, we see the enzyme cleave ATP or hydrolyze ATP, cut ATP into ADP, and that releases energy. That energy causes the protein to change shape. Now the protein no longer accommodates those 3 sodium ions. It no longer has the proper shape, the nooks, the crannies to house those 3 sodium ions. And so the way this protein changes shape is in a way that expels those 3 sodium ions from the cell. And after those 3 sodium ions are expelled, the protein has a different conformation, a new shape. And so now we're moving from number 2 to number 3 as we go from left to right. And we can see that the new shape of the protein accommodates 2 potassium ions. Just 2 potassium ions fit into that little nook. Nothing else, no other substance will fit in there. And as soon as 2 potassium ions fill that little nook in the protein, those potassium ions interact with those proteins that interact with the amino acids that make up that protein. The protein, the sodium-potassium pump, the purple protein, will change shape once again. And so now we're moving from panel 3 to panel 4. After those 2 potassium ions enter the protein and fit into those little spaces, the presence of the potassium ions causes the protein to change shape again, expelling those potassium ions on the other side of the membrane. We can summarize the sodium-potassium pump with the following bullet points. Number 1, it's a transmembrane protein. Number 2, it performs active transport. Number 3, it's present in the plasma membrane of all of your cells. Number 4, it uses ATP to expel sodium from the cell and to bring in or import potassium into the cell. We can see using this diagram that for every 1 molecule of ATP that gets spent, 3 sodium ions are pushed out of the cell and 2 potassium ions are brought into the cell. The sodium-potassium pump is responsible for generating and maintaining concentration gradients across the membrane for both sodium and potassium. And notice these two substances are both moved in an uphill fashion or against their concentration gradients. Sodium is in low concentration inside the cell, and the sodium-potassium pump moves sodium from an area of low concentration inside the cell to an area of high concentration outside the cell. The opposite is true, or maybe the same is true for potassium. Potassium ions are moved from outside the cell to inside the cell, uphill or against the concentration gradient for potassium. Cells have a higher concentration gradient of potassium on the inside. So this is a summary image. It shows us the sodium-potassium pump that we were just speaking of. And here, the sodium-potassium pump itself is represented by the red ball in the plasma membrane, the red ball or the red dot. And we can see very clearly that for every 1 ATP that gets cleaved into ADP, the cell is able to expel 3 sodium ions and import 2 potassium ions. And sodium-potassium pumps are responsible for maintaining the concentration gradients that are shown up here. The extracellular fluid has a relatively high concentration of sodium and a relatively low concentration of potassium, and the intracellular fluid has a relatively low concentration of sodium and a relatively high concentration of potassium. This is relevant for lots of reasons, but for us, primarily the maintenance of what's known as membrane potential. It's an upcoming topic, and cells maintain a membrane potential, meaning there's a difference in charge across the plasma membrane. Notice that these are charged particles. Both sodium and potassium carry a single positive charge, and they play a role in maintaining membrane potential. This information will also be relevant when we talk about nerve impulses, action potentials, graded potentials, how cells generate electrical current. The resting membrane potential, as exhibited by this distribution of ions, among other things, will be temporarily disrupted during a physiological electric current. We're still talking about mechanisms of active transport, and the sodium-potassium pump was an example of primary active transport, and it's called primary active transport because that transmembrane protein directly uses ATP in order to move a substance against its concentration gradient. So we looked at the proton pump and we looked at sodium-potassium pumps, and those are both examples of primary active transport, where the membrane protein itself acts as an ATPase. Next up is secondary active transport. Secondary active transport indirectly relies on ATP. So in secondary active transport, we need a pre-existing concentration gradient that was established by primary active transport in order for secondary active transport to occur. Here we see 2 different transmembrane proteins, and they are both involved in active transport. The membrane protein shown on the left is involved in primary active transport, and the membrane protein shown on the right is involved in secondary active transport. Let's start with what we already know, where we already understand, I hope, primary active transport. So let's start with the transmembrane protein that's on the left. This is a sodium-potassium pump. It was obviously created by a different artist, but all of the important features are represented here. So we're talking about the transmembrane protein on the left. This transmembrane protein is a pump. It is an ATPase. As a pump, it expels 3 sodium ions from the cell. So it ejects 3 sodium ions and it imports 2 potassium ions. And in the process, this membrane protein cleaves a molecule of ATP to form a molecule of ADP plus the inorganic phosphate that got chopped off. So that's primary active transport. If we look at the right side of the screen, that other transmembrane protein is engaged in secondary active transport. Notice that there's no ATP involved here directly, but rather secondary active transport depends on, relies on an existing concentration gradient that was created using ATP, but by some other transmembrane protein. So this protein is engaged in secondary active transport, but the only reason it can do its job is because there is a protein that cleaved ATP directly during primary active transport. So we'll break down this diagram into 2 halves. Once again, this transmembrane protein is engaged in primary active transport. The protein itself includes an ATPase that is able to release the energy in ATP by performing this chemical reaction. It expels 3 sodium from the cell and it imports 2 potassium inside the cell. And importantly, this gives rise to and maintains concentration gradients for both sodium and potassium. It is important to note that because of sodium-potassium pumps, outside the cell has a much higher concentration of sodium ions, and inside the cell has a much higher concentration of potassium ions. And so because primary active transport is occurring, and because that relies on ATP, we now have 2 different concentration gradients, sodium concentration gradient and potassium concentration gradient. And secondary active transport is going to rely on the energy in these existing concentration gradients to move something across the membrane. So here's the other side of that diagram. And we see this is a transmembrane protein. It's performing secondary active transport. And this type of transmembrane protein, this example here, is known as a symporter or a co-transporter. Either term is fine, and those terms show up in the next slide or the next 2 slides. So during secondary active transport, a transmembrane protein moves 2 substances in either the same direction or in opposite directions, but one of those substances is moving downhill or along its concentration gradient. So remember, there's a concentration gradient for sodium. There's a lot more sodium outside the cell than inside the cell. And remember, diffusion works in a way to disrupt a gradient. So this transmembrane protein will allow sodium to travel downhill. It will allow sodium to move from an area of high concentration to an area of low concentration. However, a glucose molecule has to move with it. The only way that sodium can move down its concentration gradient is by traveling along with a glucose molecule. So this is a cell's way, one of a cell's way of getting glucose inside. Glucose, I hope you remember, is an important resource for our cells. It's our cell's primary energy fuel for creating new molecules of ATP. And so secondary active transport is one way in which glucose can move into our cells. It's as if this transporter says, oh yeah, sodium, you want to come inside? I see you got a concentration gradient there. I see you want to move from an area of high concentration to an area of low concentration. Yeah, I'll let you through. I'll let you move downhill. But the only way you can do it is by bringing a glucose with you. All 4 of the transmembrane proteins shown here perform secondary active transport. There are 4 different examples. On the left, in panel A, we have transmembrane proteins that are called antiporters. And on the right, we have two transmembrane proteins that are called symporters. An antiporter, also known as a countertransporter, moves two substances in opposite directions. So starting on the very left, we see an antiporter that is moving sodium in one direction and calcium in the other direction. And moving over to the other antiporter, we see an example of an antiporter that's moving sodium in one direction and hydrogen in the other direction. For symporters, we see two examples. Symporters move two substances in the same direction. So we see a sodium glucose symporter and we see a sodium amino acid symporter. And all four of these transmembrane proteins are engaged in secondary active transport. Notice that ATP is not represented on the slide. None of these four transmembrane proteins are ATP aces. And by that I mean none of them are able to cleave a molecule of ATP and release the energy. Rather, they all rely upon an existing concentration gradient. And it looks like the concentration gradient in all four cases is sodium. There's more sodium outside the cell than inside the cell. So sodium is driven to diffuse into the cell, right, from an area of high concentration outside the cell. Sodium wants to, and I put quotes around that, right? An ion cannot have will or volition. Sodium wants to move into the cell. And the only way that that sodium can enter the cell is by passing through a transmembrane protein. If the transmembrane protein is an antiporter, the cell will allow sodium to enter while exporting a particular ion, in this case, either calcium or hydrogen. And for the symporters over here, both these symporters allow sodium to cross from an area of high concentration to an area of low concentration. But in order for the transport to occur, another substance must travel along with sodium. So for the glucose transporter, it's a cell's way of obtaining new molecules of glucose, or for the amino acid symporter, it's a cell's way of obtaining new molecules of amino acid. It's often helpful to look at different artistic representations of the same biological phenomenon. So we're looking at diagrams of primary and secondary active transport here. And let's start with primary active transport. In panel A, substance X is moving via primary active transport. It's being ejected from the cell. It's moving from an area of low concentration to an area of high concentration. So substance X is moving from low to high, and it's moving through a transmembrane protein here. And this transmembrane protein is also an ATPase. So if we can make up a term for this purple transmembrane protein here, we might call it an X-ATPase, or we don't know what X is. And so this is primary active transport. The transmembrane protein directly uses ATP to move one substance against its concentration gradient, as shown here. And we can contrast that with secondary active transport. So let's look at panel B. Both primary and secondary active transport are represented here. So what we were just looking at in panel A is right here. This is what we were looking at in panel A. Scoot around that S. So this is the same thing. What I just circled in panel B is primary active transport. That transmembrane protein is directly using ATP to establish a concentration gradient. And here we have a concentration gradient for X. Because of primary active transport, there's a lot of X outside the cell and very little X inside the cell. So this established gradient means that X, and I'll put air quotes here, X wants to enter the cell. Well, enter secondary active transport. This transmembrane protein, shown in gray or light blue, this transmembrane protein is performing secondary active transport. It is a symporter. It moves substance X and substance S in the same direction. It does not directly cleave ATP. Rather, it relies on the energy in the existing concentration gradient for substance X in order to move S. So the purpose of this transmembrane protein here, this guy, is to bring substance S into the cell. The way this transmembrane protein does that is by acting as a co-transporter, and it relies on this existing concentration gradient for substance X in order to move X and S into the cell. So where are we? In a previous lecture segment, we covered mechanisms of passive transport. Those are mechanisms in which the cell does not need to spend ATP in order to move a substance across the membrane. And in this segment, we have been talking about both primary and secondary active transport. For all mechanisms of active transport, the cell needs to expend ATP. Where we're going next is to talk about vesicular transport, the last bullet point shown here, or the movement of substances across membranes that are packaged in vesicles. So a vesicle is a spherical sac, and the walls of that vesicle are made out of a double layer of phospholipids, usually with proteins embedded in it. And there are two types of vesicular transport. Endocytosis brings material into a cell when a vesicle forms or buds off from the plasma membrane. And exocytosis ejects material from the cell when an existing vesicle fuses with the plasma membrane to expel the material. And in contrast to all of the transport mechanisms that we've talked about previously, endocytosis and exocytosis are big processes. It involves lots of molecules of ATP. It involves a relatively large region of the plasma membrane, and it also involves the cytoskeleton of the cell. So here's a really simple diagram of endocytosis, where a region of the plasma membrane bends and pinches off. And this requires, it's not really represented here, but it requires movement of the cytoskeleton. And so this vesicle that forms by pinching off is called an endocytic vesicle. And notice its contents. The contents of that endocytic vesicle is a mixture of the material that used to be outside the cell. So it's a mixture of extracellular fluid and whatever solutes were dissolved in that extracellular fluid. Here we have a particular type of endocytosis called phagocytosis. And what's being shown here is a cell known as a phagocyte. And it's a type of, maybe you're familiar with the term macrophage. So it's a relatively large cell. And one of its jobs is to engulf particulate material. So fragments of cells, viruses, bacteria. And so in order for phagocytosis to occur, which is a form of endocytosis, the cell extends its cytoskeleton to form what's known as a phagosome around something that's outside the cell. And then it brings into the cell that substance, and it's enclosed in that vesicle. So again, this is a very large process involving many molecules of ATP, the cytoskeleton, and the plasma membrane. Here's a scanning electron micrograph of a white blood cell surrounded by those golden rod-shaped bacteria. And so this is a phagocyte. It's part of your immune system, and one of its primary tasks is to wrap around those bacteria and bring those bacteria into the cell. And once they're inside the cell, that phagosome, or that endocytic vesicle, will fuse with another type of vesicle that's full of digestive enzymes. And so the molecules that make up the bacterial cells will be chopped up for parts. And then exocytosis is the opposite of endocytosis. Exocytosis is a cell's way of expelling material, and exocytosis is usually reserved for larger substances like proteins. And so here we see a secretory vesicle. So this is a vesicle that's full of the red dots that are something that the cell manufactured, and the cell, as part of its job, needs to expel that material into the extracellular environment. And so the secretory vesicle will fuse with the plasma membrane, and once it fuses with the plasma membrane, then exocytosis occurs as that substance leaves the cell and enters the extracellular fluid. So your pancreas is an example where exocytosis occurs. Your pancreas is part of two different organ systems. It's part of your digestive system because these, let me switch over to a different color. Let's switch over to blue. Because these acini cells here, these cells here, are all busy manufacturing digestive enzymes and the components of what are known as pancreatic juice. And the components of pancreatic juice are secreted into this duct, which eventually leads into your small intestine. There are different types of cells in the pancreas, and some of them are assembled into what are known as islets. And maybe you learn this as islets of Langerhans, or nowadays they're called pancreatic islets. There's a trend in medicine to stop naming things after people and to start giving them more functional names. So cells that are found in the islets of Langerhans, they do not secrete digestive enzymes, and these cells are not part of your digestive system. These cells are responsible for the endocrine function of the pancreas. So your pancreas plays a role as part of your digestive system, and it plays a role as part of your endocrine system. And as part of your endocrine system, beta cells, which are part of the islets, beta cells secrete insulin, and insulin is a peptide hormone. It's a small protein, and proteins, even small proteins, are too large to cross through the plasma membrane by any other mechanism other than vesicular transport. So in order for the insulin that these beta cells manufacture to get out of the cells and into your blood as a circulating hormone, these cells need to export or secrete insulin through exocytosis. I like this diagram. It comes out of your textbook. It's a lovely, very elegant summary of transport mechanisms that involve plasma membrane proteins. So most recently in this lecture, we talked about primary active transport. These are all pumps, and you can tell that they're all pumps because look at this, ATP, ATP, ATP. Primary active transport directly uses ATP to move substances against their concentration gradient. And then shown here, these are all mechanisms of secondary active transport. They all involve transmembrane proteins. Some of these transmembrane proteins are symporters, and some of them are antiporters or countertransporters. And notice that ATP does not play a direct role. Rather, all of these substances can move because of a pre-existing concentration gradient that was previously established by primary active transport. And then in the purple and in the green, those are transport mechanisms that we talked about previously. They're all mechanisms of facilitated diffusion, where facilitated diffusion is diffusion. So the substance is going to move from an area of high concentration to an area of low concentration. And in facilitated diffusion, the substance is either large or too polar or charged, and so it needs to pass through a membrane protein. So ions are obviously charged. Ions cross the plasma membrane protein in ion channels. And then glucose is passing through a transmembrane protein. Sometimes that's called a glucose transporter or a glucose carrier. So not shown here are other methods of passive transport. Notably, simple diffusion and osmosis are not represented on this image. Rather, all of the transport that's shown here occurs through a transmembrane protein. I like to include multiple diagrams created by different artists, and we can use this diagram to summarize the most salient points of transport mechanisms. And this diagram is particularly elegant, where a lot of information is conveyed using very few words and simple diagrams. So let's just go through this diagram. All of it's important, so I can't even say let's go through the most important parts of this diagram because it's so elegant. Everything on this diagram is important. So let's start by looking at concentration gradients. So for all of these transport mechanisms, concentration gradient matters. And in all three of these panels, there is a substance moving between an area of high concentration shown on the right and an area of low concentration shown on the left. So in simple diffusion is diffusion, and during diffusion, period, any kind of diffusion, a substance moves from an area of high concentration to an area of low concentration, so downhill. This is simple diffusion because the substance moves directly through the phospholipid bilayer, and we do not see a transmembrane protein here. And substances that move by simple diffusion are small, they're hydrophobic, and they are uncharged. And then we move into facilitated diffusion, and facilitated diffusion involves a transmembrane protein. And facilitated diffusion is diffusion. So a substance is going to move from an area of high concentration to an area of low concentration. And whatever this substance is, it requires a membrane protein because it's not small enough, it's not hydrophobic enough, or maybe it's charged. So substances that move by facilitated diffusion are a little bit larger, they are water soluble, not fat soluble, and or they carry a charge. And then finally in the bottom panel we have active transport, which also relies on a transmembrane protein, but look at the direction of that arrow. Active transport always moves something against its concentration gradient from an area of low concentration to an area of high concentration, and sometimes we refer to that as uphill. This diagram is so stripped down, it's so elegant, it's so simple that ATP is not shown here, but we know either directly or indirectly ATP is necessary for active transport to occur. When it's direct use of ATP, we refer to that as primary active transport, and we would call that membrane protein a pump. In secondary active transport, it's indirectly relying on a previously existing concentration gradient that was produced by active transport. So if this is secondary active transport in that bottom panel, we would call that transmembrane protein either a symporter or an antiporter. And this elegant diagram, it's so stripped down that we can't determine what it is, but what we do know is that the movement of that substance definitely requires or depends on ATP being spent by the cell. And the last item in this lecture segment is to introduce you to the concept of membrane potential. And it's very much related to concentration gradients, only now we're thinking about the distribution of charges, positive and negative charges across the plasma membrane. And so there are lots of substances in the extracellular fluid and the intracellular fluid. Lots of these solutes carry charges. Most notably, or perhaps most obviously, ions, right? Any ion is a charged particle. It carries a positive charge or a negative charge. And these charged particles are, as it turns out, not distributed evenly across the plasma membrane. And so a membrane potential exists when there's the buildup of positive charges on one side of the membrane and negative charges on the other side of the membrane. So I'm just introducing you to the topic of membrane potential. It will become very important for understanding physiological electric currents, which are important in the nervous system and the muscular system. So most cells in the body, if not all cells, have a membrane potential. And a membrane potential exists when on one side of the plasma membrane, there's a buildup of negative charges, and on the other side of the plasma membrane, there's a buildup of positive charges. And notice that for right now, I didn't specify inside the cell or outside the cell. It doesn't matter. The way we see it in this image here, in this image, we see on the inside of the plasma membrane, there are more negative charges, and on the outside of the plasma membrane, there are more positive charges, but the situation could be reversed. So just keep that in mind. And we say a membrane potential exists whenever there's a buildup of opposite charges across the plasma membrane. Membrane potential is called membrane potential because it is a form of potential energy. And I hope you remember that potential energy is the capacity to do work. So it's stored energy. And let's start with the cell on the left. And I hope you remember that negative charges attract positive charges. So negative and positive want to move toward each other, but they are being separated. They are being held apart by the plasma membrane. The plasma membrane only allows charged particles to pass through transmembrane proteins. And if those transmembrane proteins either aren't present in the membrane, or maybe those transmembrane proteins are ion channels that are closed, then those positively charged particles and those negatively charged particles cannot physically cross the membrane, and they're being held apart, generating potential energy. These particles are attracted to each other. They want to move towards each other, but they can't. So now let's apply that understanding to the dam shown on the right. So before this dam was built, this was a river. Prior to human existence, this river was flowing from an area of higher elevation to an area of lower elevation, right? Water flows downhill. And when this dam was built, it prevents and as this dam was built, because this dam was built, now the water is prevented from flowing downhill. And this gives rise to potential energy. The dam that's holding back the water is a form of potential energy. And this is what's known as a hydroelectric dam. So one of the purposes of this dam is to generate potential energy by holding the water back, by not allowing it to just naturally flow downhill. We generate a form of potential energy that we humans can now control and use that potential energy to generate a usable form of energy for us called electricity. And so in this hydroelectric dam, humans allow the dam to become permeable to water, meaning water can be permitted to flow downhill. And as that water flows downhill, the kinetic energy gets captured and is used to generate electricity. That the kinetic energy of the flowing water spins a turbine that's inside the dam, and that turbine is used to generate electricity. So the purpose of this comparison, comparing membrane potential to water being held back by a dam, is they are both forms of potential energy. There's the capacity to do work here. In the dam, that potential energy is harnessed to generate electricity, another form of energy. And the same thing is true for our cells. when we start to talk about cell signaling and the generation of action potentials, we'll see that those charges across that plasma membrane will be allowed to flow, just like water can be allowed to flow through a dam, and that's going to generate physiological electric current. We can measure membrane potential. We can physically measure the charge difference across a plasma membrane using a tiny little voltmeter. You might have a voltmeter at your home. You can go to Lowe's or the hardware store, for example, and buy a voltmeter. And you might use a voltmeter if you are interested in doing electrical work around your house. Because we're interested in electricity here, right? And cells also generate electricity, although in a different way than the electricity that you may have learned about in a previous physics course. So we can use a voltmeter to measure the charge difference across a membrane by inserting one electrode just on the inside of the plasma membrane. So the red electrode shown here punctures the plasma membrane, and the tip of that electrode resides just inside the membrane. And then the reference electrode in gray resides in the extracellular space, in the extracellular fluid. And so that voltmeter is measuring the magnitude of charge difference across that membrane. So in panel A on the left, we have the voltmeter, the cellular voltmeter that I just explained. And in the panel on the right, we have a graph that shows membrane potential over time. So on the vertical axis, and let me switch to purple here. On the vertical axis here, we have membrane potential. This is something that we can physically measure. And instead of volts, it's measured in millivolts. A millivolt is 1,000th of a volt. On the vertical axis, we have membrane potential as measured with a voltmeter. And on the horizontal axis here, we have time. And maybe that's in seconds or milliseconds, 1,000th of a second. There aren't any units shown here. So on this graph, if we start with the voltmeter where neither of the electrodes are anywhere near the cell, let's say they're both electrodes, the intracellular electrode, the red electrode, and the extracellular electrode, the reference electrode, the gray electrode, both of those are placed in the extracellular fluid at time 0, and they're kept in the extracellular fluid for this distance across the graph. The voltmeter will register no difference. There is no charge difference throughout that extracellular fluid, but as soon as the intracellular electrode is placed on the inside of the plasma membrane, that's what this asterisk indicates. And now all of a sudden we have a membrane potential as shown here. And the membrane potential for this cell is -70 millivolts. So 70 represents the magnitude of charge difference. Well, how much more positive is it on one side of the cell relative to the other side of the cell? And this magnitude is 70. And as we'll see down the road, the negative, it's just a naming convention. Negative just tells us what's going on the inside of the cell. The negative sign just says, hey, the inside of the cell happens to be negative and the outside is positive. It's a naming convention that will come up and is very important for us later. So the magnitude of the difference across this cell shown here is 70. It's -70 because in this cell shown here, the inside of the membrane is where the negative buildup is, and the outside of the membrane is where the positive buildup is. So all your body cells have a membrane potential, and for the vast majority of those cells, that membrane potential is stable, it's fixed. It doesn't really change over time. So if you look at an adipocyte or you look at a fibroblast in connective tissue, anytime you go in and you measure that membrane potential, it's the same all the time. It never changes. And so the vast majority of cells in your body have a stable resting membrane potential. A few types of cells have an unstable resting membrane potential, and these cells are neurons and muscle cells. And these cells are called electrically excitable cells, and because they can undergo dramatic changes in membrane potential that are also known as physiological electric currents. So we get to combine several topics that we've talked about previously in this slide to get us to an understanding of what an electrochemical gradient is. There are electrochemical gradients across the membranes of all of your cells. And let's think about the word electrochemical gradient. Let's start with chemical first. You know what a chemical gradient is. It just means that one substance has a higher concentration on one side of the membrane and a lower concentration on the other side of the membrane. There are chemical gradients, for example, for sodium, for potassium. There's more sodium on the outside of the membrane and less sodium on the inside of the membrane. There's less potassium outside the membrane and more potassium inside the membrane. Those are chemical gradients. We can also separately consider electrical gradients. And when we consider electrical gradients, we're only thinking about charges. It doesn't matter what the substance is. As long as it has a charge, it can contribute to an electrical gradient. So for some cells, there's a buildup of positive charges on the outside of the cell and a buildup of negative charges on the inside of the cell. And then in some circumstances, that electrical gradient can be reversed. The outside of the cell can become negative relative to the inside of the cell. So we have two types of gradients here. There's a chemical gradient when we're talking about a particular substance, and there's an electrical gradient when we're talking about the distribution of charges on either side of the membrane. Both of those gradients influence the movement of substances across the membrane. You remember that concentration gradients, chemical gradients, drive the movement of substances across a membrane. And I hope you remember, we just talked about it. The distribution of charges can drive substances across the membrane. You remember that positive and negative attract each other. So we can combine these two motive forces, these two situations that are going to influence the movements of substances across the membrane. We can combine the two concepts and call it an electrochemical gradient. And so this is a great diagram for understanding how we combine those two motive forces. So let's just go from left to right, where we are thinking about the concentration and electrical gradient from outside the cell to inside the cell. In this first panel here, we're just thinking about a concentration gradient, a pure chemical gradient. So we're not thinking about charges. There is no charge distribution across the membrane. There's no buildup of positive on one side and negative on the other side. This is just a pure concentration gradient, as like we were just starting to talk about diffusion. It's that. And so you know that substances tend to diffuse from an area of high concentration to an area of low concentration. So a chemical gradient is going to favor the movement of these blue dots in the shown direction. Now let's move on to the middle panel, where we have both a chemical gradient and a membrane potential, an electrical gradient. So the chemical gradient hasn't changed, right? We have more of these blue dots outside the cell than inside the cell. But what we've added on is this membrane potential here. And in this middle panel, we see the membrane potential consists of a buildup of positive charges on the outside of the membrane and a buildup of negative charges on the inside of the membrane. And you remember that positive and negative attract. And so look at the width of this green arrow. It's much wider now because we have the combined effects of the chemical concentration gradient and the electrical gradient both simultaneously favor the movement of these blue dots from outside the cell to inside the cell. So the motive force of the concentration gradient hasn't changed. We talked about that in the previous panel, the panel all the way on the left. What we've added here is the motive force due to membrane potential. Notice that these blue dots outside the cell, or these blue dots, carry a positive charge. And positive repel, and positive and negative attract. Notice that there's a negative charge buildup on the inside of the cell. So in addition to the concentration gradient driving the movement of these blue dots into the cell, we also have an electrical gradient driving the movement of these blue dots into the cell. So in this middle panel here, we have the combined effects of a concentration gradient, a chemical gradient, with an electrical gradient or a membrane potential. All right, let's move to the panel all the way on the left. We also have the combined effects of a chemical gradient and an electrical gradient or a membrane potential, but now the membrane potential is reversed. We still have our same old concentration gradient that we started with, but now our membrane potential exists because there's a distribution of charges along the membrane. Only here, there are more negative charges on the outside of the membrane and more positive charges on the inside of the membrane. And look at the width of this green arrow. It's narrower, right? So something must be slowing down the movement of these blue dots. And it is the electrical component of the electrochemical gradient. So the chemical gradient's exactly the same. We have a concentration gradient of blue dots that's favoring the movement of blue dots from an area of high concentration to an area of low concentration, right? But in our panel all the way on the right here, we have an electrical gradient that impedes or resists or inhibits the movement of these blue dots because these blue dots carry a positive charge and the inside of the membrane is also positively charged. And you remember that positive and positive repel each other. So there's more incentive for these positively charged blue dots to stay on this side of the membrane because there's a negative charge out here. And these blue dots are not particularly drawn to move into the cell because they are repelled by these positive charges that are inside the cell. So this is a fantastic diagram for understanding the combined effects of concentration gradient or chemical gradient, as well as an electrical gradient or membrane potential on the movement of a charged substance across the membrane, which is a huge topic when we get to cell signaling as well as the nervous system and the muscular system. For now, I just hope that you understand the concept of an electrochemical gradient. It's the combined effects of a chemical gradient and an electrical gradient, and those two gradients work together to determine the motive force of a particular charged substance across the membrane, how likely that substance is to move, how quickly that substance moves. So that brings us to the end of segment 3 of 4. And in this segment for cells and movement across the plasma membrane, we finished talking about transport mechanisms, right? We wrapped up active transport mechanisms here, and I introduced the concept of membrane potential. And that concept will be very important later on. And so in segment 4, we'll finish out this lecture topic, and I hope to see you there. Bye-bye.